Creating a human with desired traits, then fiction, now reality. Hi, this is Afrah Aneez of grade 11 AS.
This is Aan Maria of grade 11 AS, and we are from Trivandrum International School. The movie Gattaca mainly focuses on how people were categorized based on their method of reproduction. People were divided into two, valids and invalids. Valids were conceived through gene manipulation and genetic engineering, whereas invalids were conceived naturally. In the not too distant future, the protagonist, an invalid man, dreams of traveling to Titan. The categorization of people led to vast disparities. Despite all the hurdles, Vincent succeeds in achieving his dreams, taking quite the risky path.
Gene editing with CRISPR enables scientists to precisely alter, rearrange, and delete DNA of nearly any living organisms. CRISPR stands for Clustered, Regularly Interspersed, Short Palindromic Repeats. The CRISPR-Cas9 genome editing system allows for precise, efficient, and fast modification of DNA in various organisms and types of cells. The essence of CRISPR is simple. It's a way of finding a specific bit of DNA inside a cell, and the selected piece of DNA is altered. The first experiment with CRISPR-Cas9 on human embryos was done in 2015. This led to a fiery debate regarding ethical issues on the prospective clinical use of germline genome editing in humans. Despite the opposition, implementation of GGE can be expected in the future. GGE in the clinic would be coupled with IVF in order to create a genetically related child who would possess a desired trait.
Mosaicism, a situation where not all cells of an embryo have the same desired DNA modification. Off-target effects, unintended changes in the genome outside of the targeted sequence occurs. And lastly, on-target undesired modifications. An alternative GE approach has been developed and tested in human embryos, whereby, unlike in the CRISPR-Cas9 system, a sequence is not cut but a base pair is directly modified. As such, this base editing approach does not involve a repair mechanism, which could introduce undesired on-target modifications. Yet this technique is not only relatively new and not thoroughly tested, but also was shown to introduce off-target modifications.
We can distinguish the following types of embryos using GGE research based on their source: surplus embryos, which are left over after IVF procedures; embryos created specifically for the purpose of research using gametes left over from IVF; and embryos created specifically for the purpose of research using gametes procured specifically for research. One side states that human embryos have the same moral status as any other born human. Some agree that value of embryos increases during their development. And finally, the ones who support GGE, who believes that human embryos have no moral status.
A team of researchers led by Zhongzhu Huang in China injected 86 embryos and then waited 48 hours, enough time for the CRISPR-Cas9 system and the molecules that replace the missing DNA to act. Of the 86 embryos, 71 survived, 54 were tested, and 28 were successfully spliced. His team also found a surprising number of off-target mutations assumed to be introduced by the CRISPR-Cas9 complex acting on the other parts of the genome. These mutations, which were unintentional, could turn out to be harmful, Also, the rate of mutations were much higher than the mouse embryos and the adult human cells.
He Jiankui, a Chinese scientist, created embryos with sperm of a man who contracted AIDS. 2 embryos were implanted to establish a pregnancy, which resulted in the live birth of twin girls.
The argument that modifying genomes is inherently dangerous may soon end up being inconsistent. There are several risk factors in the natural way of reproduction as well.
2/3 of human embryos fail to develop successfully during the first month of pregnancy. 7.9 million children are born with genetic defects every year.
We must take prospective risks into account and research extensively in the GGE field.
Such technologies could be used to prevent genetic diseases, but there is a high probability of it being misused. Moreover, it might be only accessible to the wealthy and could create class differences between individuals based on their genetic identity.
Let's hope that we never end up in a utopian society like Gattaca, where parents and society try to fit their child into set standards,
but one which does not create a rift between the so-called valids and invalids. Thank you for watching the video.